Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
So this is Richard Jacobs with the Finding Genius Podcast. I have a good guest today, Sharmily Khanam. She's a postdoctoral research associate in the Department of Microbiology and Plant Microbiology, also associated with the laboratories of molecular anthropology and microbiome research at OU, Ohio University. So Sharmily, thanks for coming.
Thank you, Richard. It's my pleasure.
Yeah. So within the world of bacteria, are you working on antibiotic resistance or bacterial physiology? Like, what's your focus right now?
Well, I'm trained as a molecular microbiologist. My research currently focuses on understanding the evolution of antibiotic resistance in gut bacteria. So we study the impact of lifestyle choices like use of antibiotic, for example, on the microbial composition in the gut, as well as how that changes the bacteria's overall response to different antibiotics.
So, so are you looking for interventions, dietary or health interventions, or medical, like small molecule drug interventions? Like, what would be the issues in which an intervention is needed, first of all?
Well, not really. We are not doing any intervention as of right now. We are currently actually today, as much as we understand about the antibiotic resistance, we know that it has been linked to widespread use of antibiotics. So it would be interesting to see that how the microbial population in the gut of our ancestors were, and how much resistant they were to the current modern day antibiotic. So we are privileged to study a population who lives in the remote village inside the Amazon forest in Peru. And we have their fecal material, which we study to understand the composition of total microbial population that they have, and also see the genetic markers for antibiotic resistance that we know of, whether or not they are present in the gut microbial population of the hunter-gatherer population that we are studying. And
one quick question here. You said the genetic markers for antimicrobial resistance, is that the person's genes or the microbes' genes? Or both? These
are the microbial genes, yes.
Are there, I haven't asked this question, are there different populations that for some reason they have because of something in their genes, people that are able to fend off antibiotic-resistant bacteria of certain kinds, or no? Is there any correlation there at all?
So that's an active area of research. Actually, that's another dimension that I'm also interested in. So there is a very intricate relationship that happens between the host and its microbial population, meaning that what is the host eating, or how their what is their lifestyle, for example, whether they do exercise, whether they they're happy, or places they travel. All these actually impacts the overall microbiome composition inside the host, right? So within the microbial population, there are microbes who are making sure that the pathogenic microbe are in check. So definitely host behavior or host lifestyle impacts on at least controlling the infectious pathogens. So what is the direct relationship? Like how does it happen at the chemical or molecular level is still an active area of research.
Okay, well, yeah, please continue. So What's your focus? Where do you think the new knowledge is going to come from in your studies in particular?
So currently I am just studying that what is the scope and extent of antibiotic resistance in the gut microbial population, in hunter-gatherer population, which resembles our ancestors. And then comparing that to the gut microbial population and their antibiotic resistance profile in the population who are more urban or rural agricultural population, who are exposed to modern day antibiotics, gives me an idea that whether or not our ancestral microbiome was already well poised to tolerate the modern day antibiotic. So understanding that, first of all, would fill the gap of knowledge in understanding the molecular mechanism that is involved in resistance to diverse group of antibiotics, because historically our understanding of antibiotic resistance comes from mostly clinical samples or clinically relevant bacteria. So definitely understanding the ancestral microbiome would help us to fill this gap. At the same time, this will provide a foundation to investigate deeply to characterize the molecular mechanism in the bacterial population that is controlling the overall physiology and metabolism of this bacteria, and how that is related to host metabolism, and therefore what is the relationship between the host and the microbiome population that is controlling the ultimate outcome, which is the response of those bacteria to antimicrobial drugs.
Well, how are you supposed to look at hunter-gatherer populations? I mean, I would think these are people like out in the bush that never have antibiotics. They're having probably plant medicine. Are you talking about like rural versus urban, or are you truly finding populations of people that are hunter-gatherers still?
Well, we do have sample from hunter-gatherer population in the Amazon forest. So we are currently studying their fecal microbiome, and we have published. Our group has published research on composition of microbial population hunter-gatherer gut. So we do have sample from hunter-gatherer population who are naive to modern antibiotics, synthetic antibiotics. Well, definitely they do practice natural medicine. So, and antibiotics, as we all know, that we have discovered antibiotics from nature primarily. So there are natural antibiotics that is present. However, it would be curious to know whether or not those microbial population also can become resistant to synthetic antibiotics, for example, tetracycline or, the ciprofloxacin, the modern day antibiotics that are currently in use.
How do you test for that? Culturing the bacteria in their stool and exposing them to antibiotics?
Yes. So we have the fecal sample. We utilize fecal material, and we have the fecal sample, which we use to collect the total DNA, total genomic DNA, and we sequence them to identify the microbial population present in that fecal material. Therefore, we have the composition of microbial population in the hunter-gatherer population gut. And then we also can selectively culture the bacterial population and then expose them to different level of antibiotics and see whether or not they have resistance toward modern antibiotics, and also what is the extent of it. At the same time, we can sequence the whole genome of this bacteria and identify the genes which are already known to be involved in conferring resistance to antibiotics. So that's how we link that whether or not these bacteria are first resistant to antibiotics, and if so, what are the genes we find that would actually relate to the observed level of antibiotics that we are we see.
So what are you observing so far when you compare the exposure to the bacteria and the hunter-gatherers very quickly become resistant, or are they slower, or not at all?
So in my first project, we have identified that bacterial population in hunter-gatherer bacterial community in hunter-gatherer population has antibiotic resistance genes present. And when we selectively culture the bacterial population, specifically Escherichia coli, we saw that there are bacteria who can tolerate the antibiotic to the level of clinical level of resistance. So they do confer resistance. They do have means to become resistant to antibiotics, which are synthetic or industrial antibiotics.
So, okay, that was your first study. Are you seeing any patterns, or what does this tell you about the, not only, well, I guess the composition probably is very different. I wonder if it's a fair comparison to, urban populations, bacteria.
So we do see a pattern. We do identify that observed antibiotic resistance correlates to the presence of antibiotic resistance gene that we have known to be involved in antibiotic resistance in diverse bacteria. At the same time, we have also identified instances where the observed antibiotic resistance that we see, it does not directly correlate with the presence or absence of genetic marker that is linked to that resistance, meaning that we do not see a specific antibiotic resistance gene that we could relate to the observed resistance in these bacteria.
Well, when bacteria are resistant, do they just upregulate existing genes? Do they rearrange their genes and that's how they become resistant? Like, what's the origin of the antibiotic resistant genes?
Well, there are a lot of different mechanisms that is involved in antibiotic resistance in bacteria, and upregulating antibiotic resistant gene is one way of utilizing the mechanism to become resistant toward different antibiotics. For example, there are genes which can pump out the antibiotics from inside of the bacteria, from the cytoplasm. So in that cases, those transporters, those genes can be upregulated. That means they can be overexpressed in response to antibiotics, and bacteria can become resistant. The same way, there are genes which can modify either the antibiotic or the target of the antibiotic, and all these genes can be activated, upregulated, or somehow become active through regulatory processes that is present in bacteria and confer the resistance to the drug.
Okay, so again, what's the predominant way that bacteria are resistant, and is that way different in the hunter-gatherer populations versus the urban ones? Like, what have you observed?
We see a lot of transporters that is known even in clinical samples to be involved in antibiotic resistance, one of the major contributing factor. But I would not say that would be the only mechanism that would play a role in observed resistance. But that is one of the major mechanism of resistance that we observe in our in the microbial population in hunter-gatherer as well. At the same time, we also find enzymes which can modify the antibiotics or the antibiotic targets that confer and therefore confer resistance to antibiotics. So it's hard to pinpoint just one mechanism, one dominant mechanism that is involved in overall antibiotic resistance. Antibiotics are chemicals which has different conformation, and they come from different groups. They target different components in bacteria. Therefore, the bacteria as well utilize different types or different mechanisms to become resistant to those drugs. So they differ from antibiotic to antibiotic, and in some cases from bacteria to bacteria.
Well, I mean, where do you take it from here? So there doesn't seem to be a predominant mechanism. What are you hoping that this will tell you? That, I don't know, our bacteria have evolved along with us and now they're predisposed towards being resistant and they're going to get even worse? Or like, what are you learning from them?
So far we have learned that one of the knowledge that is being established in the field, and we kind of add to that information, is antibiotic resistance genes are ubiquitous in nature. We do have some idea about the extent of those genes in ancestral microbiome population through studying the hunter-gatherer gut microbiome. And in my, I believe that the research that we are conducting would give us an idea of what was the state of antibiotic resistance, what was the scope and extent of antibiotic resistance in the gut of our ancestors, and then how the lifestyle choices have impacted the evolution of those antimicrobial resistance genes in such a way that we have extremely resistant bacteria, clinically relevant bacteria that is present today. So that way, we will be able to do an intervention and manage the bacterial infection in such a way that our research would lay a foundation on understanding the selection pressure that has led to current day antibiotic resistance problem. And therefore, it would allow us to do an intervention and find where we can make that intervention to manage the antibiotic-resistant bacterial infections.
Well, yeah, I would think that naive microbes would, they could either become resistant as quickly as non-naive ones or less quickly, which is probably what I would suspect. I don't think it would be more quickly, but again, what does that tell you? Unless the mechanism is different, I don't know. How do you think you could find cures or remedies or new ways to attack current bacteria? Maybe there's a novel way that naive bacteria form antibiotic resistance. Maybe that gives you a clue. I don't know. And how do you imagine that you'll find a novel way to defend against AR? So
the antibiotic resistant genes are present everywhere in the environment or in the host environment, in the natural environment or in the host environment. We know that by now. However, what is the source of those antibiotic resistance? Whether or not they are always present in pathogenic bacteria. If not, then who are the carriers of those genes and what makes them to transfer those genes to the pathogenic bacteria? What kind of selection pressure is mediating that transfer? What is the molecular mechanism of those transferring those genes? These are essential questions that we need to understand in order to make that intervention. Because if we understand that who contributes to this transfer and what are the selection pressure, it would be easy to intervene the process of transferring those genes to pathogenic bacteria. Because
supposedly they get the resistance genes from other bacteria that have them, right? And bacteria, I would think, exchange through plasmids and other means all the time. And the pressure supposedly is not wanting to die. It's not the case.
Yes, you could say that. We don't know the whole story yet. We have some insight into the transfer mechanism. Yes, we do. But when we are studying an ancestral gut, we get to see that whether or not these genes were, present in some bacteria that, those are not pathogenic at all. And they just carry these genes. And when there is a selection pressure, then that's when they transfer these genes to the pathogenic bacteria. And
I mean, isn't there a library of bacterial genes? I would think there would be. And let's say it's an efflux pump, that's associated with some kinds of resistance. Have people looked through the library of bacteria genes to look for efflux pumps across a whole bunch of strains and species and bacterial types to find them? And maybe then you can do like a phylogeny of the efflux pump, for instance.
Yes, that's true. So the efflux pumps are not necessarily, actually, they are not dedicated and antibiotic resistance genes. They are part of the bacteria for the regular bacterial physiological processes, like getting rid of toxic metabolites from the bacteria. So they were present in bacteria from well before the antibiotic was discovered, or well before we could industrialize our use of antibiotics, right? So the question that we ask is in what situation these efflux pumps have been dominantly became involved in extruding antibiotic. Is it only antibiotic or other lifestyle choices that may contribute in diverting the focus of these efflux pumps from just extruding the toxic metabolites to extruding the antibiotics that the bacteria is exposed to?
Well, yeah, right. Efflux pumps are one thing, but what's another type of mechanism for resistance?
So that
is there hardening of the bacterial wall, or what?
The antibiotic resistant, the antibiotic modifying enzymes are the other major contributor in antibiotic resistance. For example, the current day beta-lactam resistance that we see in a lot of bacterial pathogen, it is due to the beta-lactamases and evolving new category of beta-lactamases that make the bacteria more and more resistant toward all these antibiotics. So antibiotic modifying enzymes are one of the major contributor in antibiotic resistance of bacteria.
Okay, well, so do you feel like you're getting close and understanding at least the origin of some of the resistance genes? I mean, what indicators are you getting right now?
So it's our understanding of genes involved in antibiotic resistance has been, advanced quite a bit. However, through my study, what I have gathered is we do lack the understanding of molecular mechanisms that is involved in antibiotic resistance in bacterial population that is in the population in an environment that is unexposed to antibiotics. So in another way, our investigation and understanding of molecular mechanisms of antibiotic is heavily relied on clinical samples or studying clinically relevant model organisms, which are pathogenic. So when I did my study, I have discovered that when we do an alignment of observed resistance and the genomic markers, which are already known to be involved in resistance to that particular group of antibiotic, we do see discordance between the genomic marker and the observed resistance for several antibiotics. So that tells us that we still need to understand the molecular mechanism. We need to include the microbial population who are naive to modern. That way we can fill the gap in knowledge and have an unbiased look at the overall antimicrobial resistance gene presence in a defined niche.
Well, I mean, it's not really the niche if you're including bacteria that really aren't present in most of people now. But it still would tell you, I'm sure, a lot about prevalence of these genes regardless in background population.
Well, not necessarily. We are studying the Escherichia coli population. So we are actually focusing on the bacteria that is clinically relevant and has been studied extensively in clinical setting. Therefore, it's comparable. Then we can compare the genomic background of the bacteria that we are finding from an antibiotic naive population and also the information that is available for antibiotic exposed industrial population. That way we can have a comparative view of genotype and phenotype concordance and discordance for antibiotic resistance in this bacteria.
Well, very good. What's the best way for people to find out more about your research and to ask questions? Where can they go?
Well, I have my LinkedIn profile or my email address, and they can definitely shoot me an email. I have the Google Scholar account where I have my published research available. And then I work with a multidisciplinary research group, Laboratory of Molecular Anthropology and Microbiome Research. So they are a research group that is present here at University of Oklahoma, and they have their website. And people can definitely look into our group's research through the web pages and the current, discoveries or publications that comes out, or reach out to my PIs or me about the research that we do.
Okay, very good. Well, Sharmily, thanks for coming. I know it's a complicated topic, but I appreciate you explaining it, so thank you.
Thank you so much.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.